Electrofusion involves application of short high-voltage electric pulses to cells in close contact. In this video, we demonstrate electrofusion of cells in vitro by means of modified adherence method. Experiments were performed on mouse melanoma cells B16F1. For the experiment, follow these steps. Grow cells in 2 separate culture flasks to 80% confluence. Separately add 2.1 microliters of each stock solution, 10 millimolar CMFDA or CMRA respectively, to 3 milliliters of Krebs-Hepes buffer in a centrifuge tube. Rinse cells twice with Krebs-Hepes buffer and then insert loading solutions into the flasks. Loading solutions contain approximately 7 micromolar CMFDA or CMRA respectively. Incubate cells for 30 minutes in a controlled atmosphere. During this first incubation, reagents pass freely through cell membranes into the cytosol, where they are transformed into membrane-impermeant reaction products. After this first incubation, rinse and then incubate the cells with culture medium for another 2 hours. The cells are observed using a fluorescence microscope equipped with a cooled CCD camera. In the acquisition software, set the excitation wavelength to 548 nanometers and choose an appropriate bandpass filter for red fluorescence image of the cells loaded with CMRA. For the cells loaded with CMFDA, set the excitation wavelength to 492 nanometers and choose a bandpass filter for green fluorescence image. Trypsinize and